Salt makes your dinner taste great, right? Well, it does, but it also has the ability to kill your food. Take Arachis hypogea, better known as peanuts. Without intervention, this iconic salty bar snack wouldn't even make it out of the ground, let alone into your mouth due to soil salinity. So, we genetically modify them. Scientists add in new genes that encode special machinery within the cells to maintain homeostatic levels of salt. This is required due to the glycophytic properties of peanut plants, meaning they grow ideally in non-saline conditions. Welcome to the genetic basis of stuffing things. Soil salinity is a detrimental environmental stress that is caused by high concentrations of sodium ions. This causes plant roots to experience osmotic stress due to sodium influx into the cells, inhibiting the plant's ability to absorb water from the soil. Plants are most vulnerable to the effects of soil salinity during their seedling and juvenile stages, as the sodium disrupts physiological processes and can break down and reduce enzymatic activity within the cell's cytosol, disrupting normal plant growth. This causes symptoms such as leaf burning, or in extreme cases, death of the entire organism. Sodium-hydrogen exchanges are protein-coding genes that aid plants in maintaining ionic homeostasis when under osmotic or ionic stress. They are a type of antiporter that appear in plants that are capable of pumping sodium ions out of the cytosol, allowing the continuation of processes needed for the plant's survival. In 1976, scientists found NHX antiporters for the first time in plants while doing research on barley root tips. Research continued, and in 1999, while researching these antiporters in Saccharomyces cerevisiae, better known as yeast, researchers from Massachusetts Institute of Technology discovered the NHX gene within Arabidopsis thaliana. At this time, the genome was only partially sequenced. Yet the research group was able to identify this gene and determine that in the presence of sodium ions, expression of the gene increased. After the initial discovery, a family of 8 AT-NHX genes have been discovered, 4 of which encode antiporters on vacuoles, 2 on endosomes, and 2 on the cell's plasma membrane. In 2012, a group from Texas Tech University investigated the expression of AT-NHX1 in peanut plants to improve salt tolerance. The AT-NHX1 gene was introduced to Agrobacterium tumefaciens in vitro and used to perform Agrobacterium-mediated transformation with explants of mature peanut plants. This allowed the AT-NHX1 gene to integrate into the peanut genome and be inherited through cell divisions, creating happy and healthy transgenic plants. The AT-NHX1 gene encodes for a vacuolar sodium proton antiporter that, alongside various other physiological conditions such as cytosolic pH, potassium homeostasis, vesicle movement, and protein targeting, increase salt tolerance. These conditions initiate the flux of sodium ions into the vacuole, moving in the opposite direction to protons across the membrane. The movement of sodium ions ensures the cytosolic enzymes do not degrade and hence do not disrupt cellular processes needed for peanut survival. The gene was successfully transformed into the genome in 11 of the 12 putative transgenic lines, a considerable success rate. These root shoots were transferred to a greenhouse and sodium chloride was added in increasing concentrations to test the effectiveness of the gene. Phenotypic differences were clear between wild-type and AT-NHX1 expressing plants after 150 millimolar of sodium chloride treatment. Transgenic plants grew larger and healthier than wild types, which showed significant chlorosis due to enzyme degrading. Additionally, the transgenic lines were discovered to have greater fresh root and shoot biomasses than the wild types under saline conditions. This is believed to be due to the transgenic plants having greater chlorophyll content, photosynthetic rate, and photosynthetic surface area, which enabled the plants to develop more successfully than wild types. The sodium being pumped through the antiporter and into vacuoles allowed survival of cytosolic enzymes, and hence, survival of the peanuts. This research demonstrated the success of genetically modifying peanut plants with the AT-NHX1 gene to improve salt tolerance. But are they on the market? Not yet, but more research is being conducted to produce more profound lines before mass cultivation is approved. For example, research has been carried out in various other genetically modified species, such as cotton, soybean, and potatoes, to consider the salt-tolerant properties further. Other studies indicate that various other transgenes would improve salt tolerance further when in combination with AT-NHX1, allowing the story of salt-tolerant peanuts to continue until we figure out how to save a peanut's life with genetic modification.